Alright, here we have Eugene, and in Eugene I'm going to start off by making some dot plots with some GPX genes, just to give you an idea on how to do it. So I go down here, I go to Tools, Build Dot Plot, and I'm going to go search for my FASTA files. So I'm going to first, I know I've stored them on my desktop in a particular folder, so let's go find that. And here I have GPX1, so these are 3 different GPX1 genes from different organisms: chimps, Homo sapiens, and mouse. So I'm basically going to compare, let's say, chimp and Homo sapiens. Now this one should be very similar. We might get some surprises here, but because I've chosen some slightly different things that it may not show up as well as you might think. Oh, I did chimp twice. Let's do Homo sapien. There we go. Okay, so we have all of this. Leave everything as it is. Now, what are we going to search for inverted repeats? We could. That's always a way to get more information, but we're going to leave it off here. Let's see what their recommended base pair is for. I'm going to increase that a little bit. I'm going to go to 6, and we're going to reduce this down to about 80%. Let's see, 100 divided by 6, 25. Yeah, about 80% should do something. And I'm going to look at it. It may be too much, but that's how you start with these things. Yes. Alright, so here I'm getting a really nice picture, alright, in the sense that there's a nice diagonal there. That's too much. We want it more sensitive. We want to find the places where it's very similar. So we want to what? We want to go and set our parameters to be more stringent. So I'm going to increase this to about 10. Alright, and then I'm going to increase this to about 90. And we'll see what we get. Okay, what this is telling us is that the human and the chimp are almost identical. I mean, it's really, really similar. There's almost no difference between the human and the chimp, and you would expect that. And that's what this diagonal does. So let's do this, and let's make it as stringent as possible, which means you make this 100. Alright, and you can reduce this size back down. Like it said, it started at 4. Let's see now how well it does. Alright, well, we see a lot. It's very stringent, but still you can see that we're getting a nice diagonal. There are a few gaps. Alright, and let's change this again. So now let's say it has to be, let's say 14. Still just incredibly similar. So we go parameters, and we go something like 34. Still just incredibly similar, almost no differences. This is not useful in the sense of finding the interesting pieces of GPX, because the two organisms are too similar. You don't get any type, there's no time for evolution to change the sequence, so you weren't able to figure out which sequences were more important than others because there was no time for it to change. So now let's try a different approach. Alright, so I'm showing you how this fails. Alright, by the way, if you wanted to see the sequence that's here, you click on that and realize down here in the highlighted box, to see this here or here, and if you wanted to copy that sequence and make a FASTA, you can right click, alright, and you can select the sequence region, or you can copy, alright, copy selected sequence, or you can do the complement, but copy that, or you could find the amino acids. You can copy the amino acids right here and make your amino acids directly from this. In other words, what amino acids does this translate to? So there are lots of things you can do here, but the idea here is right now we just want to copy the sequence. If we wanted, we would go here. If we wanted to get the amino acids that translates into, we would go here. And these are RNAs, so you would go straight to amino acids the way I showed it. So you would go here and here. If this were DNA, you would go here and here to find the RNA and then the amino acids that that RNA translated to. But the way this is set up, we would go here and here. Okay, so moving on to something that might be more useful, we have to use some sequences that are a bit more distant in evolutionary time. So we can just make this one go away. This one is not useful to us. No, I don't need to save that. So we're going to go and we're going to go do dot plot again, and we are going to go to human, right, as our first one, and then we're going to go to mouse for our second one. Alright, and we will not do inverted repeats, and we'll start where we ended last time, which I think was like 34 and 100, and let's see if we get anything at all with this really stringent well, actually we did. I was expecting to get 0 here, so this means this is extraordinarily conserved, this piece right here. So everybody can copy this. This is probably conserved across every species, but as you can see, you have a piece of the mRNA that is conserved regardless of the distance here, it looks like, because that's extraordinarily conserved. Is it 100%? I don't know. It might be. AAGAGTTCAAGGT. Yes, it is 100%, which means your consensus would be this piece. So this one is conserved all the way through all the GPX1s from mouse to human, and that's quite a bit of distance in terms of let's see, let me change the way this looks. Don't like any of those. Alright, well, what we're going to do is I'm going to reduce those stringencies. Actually, I'll do the whole thing over here. I'm going to go between human. Alright, again. And I'm going to go with mouse. Okay. And we're going to 34 and 100 actually showed something. We're going to reduce that just a little. We'll come down to say maybe 24, and I'm going to come down to maybe 95. Alright, so just a little. We were at 32 and 100 and we got something, which was surprising to me that it was that conserved between human and mouse. But we want a little bit more than that. And there, I reduced it, and now we have 2 pieces. So here we have 2 pieces that we can look at. Alright, let me scroll up. Now I'm beginning to see how this works. Not going to scroll for me. Changed something I shouldn't have. Yep. Well, we'll do this again, won't hurt. So we did build dot plot, Homo sapiens, and mouse. Alright, next. Alright, and we did 24 and 90. Okay, and actually I must have changed something because we now have 1, 2, 3, 4 pieces of interest. We would copy those and try to find these inside our other species. I've just lost my, I hit something down here that lost my, I think they're right there. And I can't seem to, there we go. Alright. Now let's raise this back up. Okay. So if I want to copy this piece here, I go down here to this one. Now this is showing me my amino acid. I could copy the amino acid by going copy and paste. And so copy selected sequence will copy the amino acids. Alright, and you would put that into a FASTA. So we would go over here, you know, to, you would go to Notepad. I have a text editor here, the equivalent, and I can paste that in. Alright, and here would be a very small sequence that I can put in there. So that would be conserved between Homo sapiens and mouse, and maybe I would find the location here. I'm just going to count these as 1, 2, 3, 4. Bring that back. Well, I'm going to call this number 4, but I need to say what parameters I used. I used 24, and you have to create your own nomenclature to tell you what your search was, and 90. No, 95. Or did I do 90? I think I did 90. That's why I got a little bit more. So that would be the first one here. I'm going to do another one, and I'm going to try and find where that compares with the others, maybe in the same region. So let's try that. Let's try doing it with chimp and see if we can find the chimp version of this piece. So this was human, and the human was at 9. Let's see, these are, human is along this way, so that's at around 500 to 570. Right? No, let's see. 600. Okay, in the chimp it's 600, and in the human it looks to be about 500. Alright, so if I do another dot plot, we'll close this one. I'm looking for about 500 in human. Alright, and so let's look at the chimp. We can build a dot plot between Homo sapiens and the chimp. Of course, these are very similar. Alright, and we just have, I'm going to make it like 50 this time, and 100%, and we still get, it's just automatic here that it's going to do it. So Homo sapiens are along here, and we're looking for around 500. So the piece between chimp and human, around 500, is, what is it in chimp? Because we want the chimp version, not the human version. So let's scroll. Let's see. Here we have the chimp version, and up here we have the human version. So we want the human version that's around 500, and that's right in here. So human was around 500. Let's do it this way. Yeah, it starts right in here. So what we would like we would like something around the, what's the equivalent region down here in chimp. It starts at about what is that, around 170. Alright, so we can just copy this whole thing in the chimp. Alright, it's going to be very big, but alright, and we're going to put that into our text editor. Oh, I was doing amino acids, so let's do the amino acid version. Oops. Alright, amino acid, and we're going to copy the amino acids, and we're going to put this. Now this is going to be huge. Alright, at least compared to this one. But what we need to do is we need to align these, and actually we can do this by hand. We can see LP, LR, LP, is there LPG, LP, LVG, alright, we'll let the computer try to find where these are similar, if it can. So this is going to be Homo sapiens and chimp. Alright, and this would be number 2. And I think we were doing like 50 and 100%. Alright, so I'm gonna take this, I'm gonna save this. I'm going to go, just the way I would name it is, this is Homo sapiens, and well, I'll use a big X, means kind of like times, and I'm going to say mouse and time, or big X times chimp, kind of to tell me that's what I was comparing there. I'm going to put this on my desktop here. Save. Alright, and we'll just close this. Now what I need to do is I need to go align those two and to see if they are even alignable. So for me, I'm going to we're going to go down here, let's see, where is it? Multiple sequence alignment, and I'll go do let's do, you can do any of these. I kind of like K-align, but let's do muscle. Let's do muscle. So I'm going to put my input file as on my desktop here. Where did I put this? Did I remember to put? Oh, here we go. Here it is. All right, we're going to open this one. Okay, now the output file, I'll also put it in the same place. Let me just copy this one. I'm going to copy the whole, there we go. Alright, but I want to call this .aln. That'll be my alignment. So I'm going to save this, and we don't have to do, we're already in amino acids, so we don't have to range it. I believe muscle will do amino acids and not just DNA, because it's asking alignment. So let's try this. You can look down here, and yeah, it did it just fine. And so there's your alignment. Now you're going like, that doesn't look like any alignment to me. Well, the funny thing, it isn't. These things are horrible at aligning. That's one of the reasons I wanted you to do this, is for you to align it. I see up here LPGT. So is there anything that looks like an L? Let's find an L here. L, skip 1G. Alright, that looks good. L, skip 1G. So L, skip 1G. And then a TLR. So we L to, well, here's an L. The thing is, these are supposed to be in the same region, and they don't translate very well. Getting a bad result is okay here, alright? This is showing us maybe that this is not the same regions. These shouldn't be grouped together. This was simply the best, the piece that aligned the best according to, that matched the best. It doesn't mean it was the best. Here's a W in, this is horrible. This one, I'm just showing you that the programs don't work very well. That was actually one of my main points to show you, is that just how poorly these alignment programs work, and they do. Unless you kind of do it by eye, I wouldn't take them as very reliable. But if you do find two regions that are pretty close to one another, this is a decent way to do it. Now, a better way to do this is to take one of our sequences, like this, the top one here, and so that's what I'm going to do. I'm going to take that one. I'm not going to save that project. I'm going to go back up here, and I'm going to grab where nope. I'm going to grab that little piece that we found. Let's see. Where was it? The alignment. GPX1s. Oh, here we go. I'm going to take the smaller one of these. Alright, and I am going to, oops, I'm going to grab this one. And I'm going to go copy. Alright, and we'd like to know what it's similar to and where. And to do that, we're going to use blast. So the good thing about this is that we know these two line up from the dot plot. The dot plot is always your best tool, and it was a small piece, and it was conserved from mouse to human. This is an important piece if it's conserved that far, so that's the way you can look at it. Comparing between human and chimp, it isn't actually very useful here because they're too similar. You don't get any reduction in the size, but human and mouse is pretty good. So I'm going to go to blast P. Alright, we'll go here, and I'm going to enter in my sequence here, and I am going to look for, let's say a particular species. Oh, maybe I won't, because blast P doesn't give you much to choose at this point. Let's just blast it and see what we get. We can get several species. You can search for the one you want after we get the sequence here. What is it saying here? Oh, we're putting in two. I see my problem. I just want blast. Let's just do blast. I was trying to align them. That's different from just a regular blast. I hit the alignment one. Alright, so here we put in our sequence. Alright, and we put in our organism. So let's say your organism was, let's do something nobody had. I don't think we gave rat. No, let's choose something a little different than that. What's a good organism? Shrimp. Something in the water, maybe. Something really distant. Let's see if there's a trout. Yeah. Coral trout. Okay, so that's pretty distant. Alright, and let's see if this is conserved that far. And I will search. And it is searching. And we got something. Alright, now it's not great. 1.7 is not a good e-value, but it found NADH dehydrogenase, and that is a similar protein. You wouldn't know that, but that's what you could report if trout were your organism, were the organism I gave you, and you would report that, hey, this actually has some similarities, the structure to it, to something, another enzyme called NADH dehydrogenase, and there may be some relationship between the two. Maybe they do something different. I mean, something similar, and actually that is true. So all of these here, none of these are great, all right, but there is some similarity between NADH dehydrogenase and GPX1, and it may be that GPX genes evolved from dehydrogenase genes. But we can go to the graphic summary. You can see the places where they are similar. So let's move back a little. All right, let's go back to our original search, and let's search for an organism that is, say, a little more similar than trout. Let's do bonobo. Bonobo should be very similar to humans and chimps. Some people say that bonobos are closer to humans than chimps. No, let me think. I'm trying to find bonobo. I don't. Spell it right. There we go. Bonobo. All right. So let's do this. Okay, even though it adjusted it for us. But what did it come up with? We still didn't get GPX. This little sequence of proteins, what we have here is I should say amino acids. It's a sequence of amino acids that's used in many proteins. So the thing is, these proteins they're little pieces. They evolved from one another. And there are some sequences that are common to many different proteins, and that's what one of these is. Now, I happen to know that the other one, the NADH dehydrogenase, is one of a family that GPX is thought to be evolved from. But you can see here, we don't see anything here that is GPX. All right, but we do see relationships, and we can learn about the biochemistry that's similar between these proteins, all right, and the one we found. Now, we're not finding any GPX here directly, but we're finding all these others, and all of these others have at least a piece of the protein that does something similar to GPX at that level. But notice here that the E value is still not overwhelmingly great, all right, and that is a bit worrisome, all right, but you have to just write that into your report and say, hey, the E value isn't always the end all, it's what you're shooting for, but with a short sequence, you often don't get a very small E value. DNA helicase, really interesting that it came up with that one, all right, but you can download some of these places and report on them. So realize this is I am not grading you on getting everything exactly right. It's on finding these conserved regions and then trying to figure out and interpret the meaning behind what you found. And that only comes with practice, and that's why I'm doing this video, to show you kind of an opposite where it was almost too similar, all right? So similar, and then one where you found nothing. So it was they were two organisms that were too far apart, and then one where the two proteins where it has parts that are too similar. So you have all of these problems. You can explain these and use these concepts in your paper for explaining whatever results you get. If you don't get anything, there is an answer. And in this video, I gave you some of the answers that you can say, hey, I didn't find anything because these organisms were, say, too far apart. But you could also make the argument that, oh, I didn't find anything because this this sequence is used all over the place, which is kind of a weird way of saying it, but it's used all over the place, and so I'm going to find too many similarities out there for this little piece. This little piece is too similar to everything because all of these proteins all evolved from a single protein sometime in evolutionary history, and this piece was so conserved that it's just everywhere. Now it's a little different, and you don't get a big E-value, but that's because this piece is found everywhere. So it's a really tough thing to do, and that's one of the things I wanted to explain to you is that you have to have some context for doing these things, and sometimes you don't get what you expect. Sometimes the E-values are bad, and you have to actually have extra knowledge sometimes to do this. This is why computer programs can't do a lot of these things for you. You have to do it by hand, and unfortunately, paper after paper that I read, they're trying to just automate it, and then they just take what it says for granted. And as you can see here, there's so much involved with interpreting these that this is a really poor way to do research. So you have to know something about your sequence. The bioinformatics just adds, it can help you find some things and some clues, but you have to have kind of a path, a direction. This can be used as a compass, but you still have to have a destination in order to use your compass. So you can think of it that way, as bioinformatics usually won't solve your problem. It won't get you where you want to go. You kind of have to have a direction, and then you use your compass to head in that direction, but you've got to know where you need to go in the first place. All right, that's this first lecture, and hopefully this will help you on the projects.